We're honored to have here today our speaker, Dr. Jianming Yu. Um, Dr. Yu is currently the Pioneer Distinguished Chair in Maize Breeding at the Department of Agronomy at Iowa State University. Dr. Yu's research tries to integrate knowledge in quantitative genetics, plant breeding, genomics, molecular genetics, and statistics to develop and apply new strategies and methods for trait dissection and crop improvement. This afternoon, Dr. Yu will talk to us about the critical role of frequency in genetics, genomics, and breeding. He'll try to talk about many opportunities and challenges in genetic and genomic analysis of qualitative and quantitative traits that are associated with frequency issue. So let's all welcome Dr. Jianming Yu.
Supposed to hear clouds, right? Thank you very much. Thank you, everyone. Yeah, it's a great opportunity to speak to this group, and I always, envious about whatever the wheat cap and barley cap has been doing. I feel you guys have a sense of a community. I think within our corn community, we'll try to do something also together. Research, grant money is one thing, but have this interaction is very important here. And I know some of the TCAP members. I see Jamie was there. That's nice. As I talked to Jeff, feel free to raise your hand through questions because I see we have 26 of us. It's okay. I only have 30 slides, but the real, information packaged, high density, but we can go through slowly. Okay. I want to start this take-home message, and then I'll repeat this one at the end of the presentation. So it's allele frequency and genotype frequency are fundamental concepts in genetics and plant breeding. We all pretty much aware breeding is a number game. That's why, in order to get a few good elite by elite recommendations, so the good inbreds, you have to play the number game. And, the reason is that because allele frequency and genotype frequency. You can see almost say that plant breeders always try to increase the frequency of the good alleles and try to purge the bad alleles. And in terms of cultivars or inbreds or hybrid, it became a genotype frequency. And then many of the opportunities and the challenges in current genetic and genomic analysis and breeding of quantitative and qualitative traits are associated with the frequency issues, which I'll use 4 different areas to explain. Let's review the basic genetics first before we go into this other published research or ongoing research. We all know that the Mendel's first law of segregation. Somebody was moving the slides. Okay, let's say here. Law of segregation, sometimes law of equal segregation, is basically saying that if you cross a mutant with a wild type, and control this gene control the single gene control one single trait, the F1 is going to segregate during the gamete formation, 50 big allele, 50 small allele. There's no viability issues. Everything is equal. And then the random formation of the gametes together became F2 individuals, where you get this 1/4, 1/2, and 1/4 ratio at the genotypic level. And then at the phenotypic level, depending on the dominance or additive or effect, you might see 3:1 ratio, this frequency. So the father of genetics was playing with this frequency issues, right? Allele frequency and genotype frequency. And if you actually learn some of the quantitative genetics, classes, you understand for a population in Hardy-Weinberg equilibrium, you can calculate the additive variance, which is a VA, variance of additive effect together is equals to PQ alpha square. P is and the Q are the allele frequency, 2 alleles, and together is 1. P plus Q equals 1. Alpha square, alpha is the average effect of allele substitution. And then it's equals 2pq a plus q minus p in d and squared. So I purposely not highlight these formulas. Do not get anybody scared of these formulas. But we have to use this to deliver our concept, okay? And the dominance variance is 4 times p squared, q squared, and d squared. As you can say, because you have so many squares for dominance effect, by default, dominance variation is going to be small, right? Because of, p and q are all smaller than 1. And if we do QTL mapping, you all understand that when we do this QTL mapping, the basic formula for the average difference between the two marker class, big M, small m, under F2 population is 2A, 1 minus 2R. So 2A is actually equals big A, small a, is big Q, small q. That's, in classes I'm teaching, I try to encourage students to derive these formulas. The 2A is the distance between the two homozygous parents, that, a homozygous genotype. And the Y minus 2R is the recombinations happened between the M, which is marker, and the QTL. The frequency come into play. And if you remember anything about this linkage disequilibrium or association mapping, you know this D equals PAB minus PA times PB. So it's basically the gamete frequency, AB frequency, minus a product of allele frequency, PAN and PB. And then it turns out if you use another more relevant measurement, which is R square, it is the square of the correlation coefficient of these two at these two loci. So d squared divided by the product of these allele frequencies. So the frequency does apply here. Okay, moving to slide number 4. And today's talk will show some examples of frequency issues. The first example is actually not our study. It's actually a publication in Nature Biotechnology. But I found that's probably a good homework question or good practice for grad students to practice their frequencies. Mute basic genetics, MutMap. And second study is some study we've done recently about the genome-wide association studies, GWAS. And then the third study is a QTL cloning process. We're going to show you with the basic qualitative traits, QTL cloning, multiple haplotype, and the frequency issues there. And the last one is the missing heritability study, a rare variant. That is a study we've been in collaboration with some of a group of human geneticists. You can see also what is a rare variant's contribution to the missing heritability issue. So MutMap is a paper published in Nature Biotechnology in 2012. I found this kind of interesting. The paper, I think Jeff has uploaded this into the Adobe Connect. The title is Genome Sequencing Reveals Agronomically Important Loci in Rice Using MutMap. Before we start this topic, I have to warn everybody that maybe one of the common comments I get is, oh, they won't work in maize or won't work in wheat, but nobody will make it work for your crop of your species of interest. There's some thing that you can learn from it. You try to adapt to any complex genomes, right? Because they're not supposed to make it useful for us, which is maize or barley or wheat. That is a much complex genome. But in rice, they can do this, which is nice. And also, there's a slight distortion of the slides, but we'll explain that next. So what they did is actually to identify a EMS mutant and cross to the original wild type, and then do a bulk segregant analysis type of analysis. But what they did is actually whole genome sequencing of pooled DNA of F2 plants showing this mutant phenotype. We have a better slides. This is just some highlights. And then they calculate what they call the SNP index based on sequencing reads. And this index is used to map or pinpoint the causal SNP underlying mutations. This might be also a great opportunity to resharpen your skills on calculated frequency or thinking or formulating questions in terms of basic genetics and frequencies. So here's the scheme. On the, let me try to have, okay, have the pointer here. Okay, this individual on the upper left is the original wild type individual. This one has the full genome sequence available. So that's the first assumption or first condition first. And then they did a EMS mutation, mutagenesis, and then they found this mutant. In this case, obviously, it's not agronomically beneficial, right? Unless you want to looking for wheat plant. But just to show this example, you could actually change this to much bigger plant or vigorous plant. This mutants actually you have observable phenotype. In the previous bulk segregation analysis and show map and other next generation mapping, what you would cross this mutant under this background to a different ecotypes or different cultivars. You want to rely on the difference between the different genetic background for the markers. But in this scheme, it looks so different, counterintuitive. They actually cross these mutants to the wild type parental line itself. So this is something I want to focus. This is really different strategy. You cross this mutant with wild type parental line itself. You get F1, you get F2. Within the F2, and you pick those individuals showing the mutant phenotype. You actually bulk, extracted the DNAs together, you bulk them, then you sequence. Okay. Interestingly, that when you do a EMS mutagenesis, the single mutant individual, does not have only single SNPs. It have multiple SNPs. So when you pull this DNA together, you sequence, you align the sequence to the wild type parental line. This happened to be a line that has full genome sequence. You find actually lots of SNPs. They said, I think the paper they reported the average you get 1,000 SNPs across a genome. But because here you actually bulk this F2 with mutant phenotype together, then you can calculate the SNP index. Imagine that one of the SNPs is actually, sorry, I'll try to move the pointer here. Let's say this SNP is a causal SNP, underlying mutations, right? You imagine if you bulk F2 progeny showing the mutant phenotype together, and then this wild type sequence is a T on the right side of the columns, and then all the sequence reads actually from mutant pool should be all Gs. So you get a SNP index of 7 out of 7. So the G, the mutant phenotype on the top, and then the all sequence read on the bottom, so it's 1. But you imagine that there's another SNP on another diff chromosome, right? That has nothing to do with this mutant phenotype. Because you're bulking multiple individuals, they could have a C or could have a G. So actually expectation of this SNPs is half-half, is about 50%, or here in this case, you can have a 3 out of 7. So it's actually at least slightly is approaching this 0.5 frequencies. And then, so here in this frequency in MutMap, which if you are interested, you can actually look at the original publication. Here, I only list the 3 relevant questions and provide some of the derivations, 2 of them. So the first question you can ask, what should be the sequence depths, right? This seems like to be out of blue. Why are you asking this question? You can formulate this sequence depths or in terms of read depths, and because you have to average them together, this actually it's a binomial distribution of random SNPs. So it's either G or either C. So the probability of having a random SNP with a frequency of sorry, it's hard to move this pointer. So this random SNP has a frequency of one half. So let's say you have 10 reads. What is the probability that this random SNP will also get an index of 1, meaning that all 10 reads will be G, which is the mutant ones. And then you can calculate that is actually the 1/2 raised to the 10th power is about 10 to minus 3. That's very rare. So this actually can come into your consideration when you're planning out the experiment. And also another question would be to simply ask, how many F2 individuals do I have to group? This F2 individual, meaning that, remember earlier, if you find some mutation, you have to self them to show this homozygous recessive mutation. Also, this has to be a recessive mutation. And how many F2 individuals to ensure that I can observe at least one mutant? It turns out that the probability of because the small e is one quarter, remember that basic genetics. And then if I have 20 F2 individuals, you have 20 F2 individuals, if I group 20 of them, the probability having at least one or more mutant is actually very high. It's close to 1, it's 0.997. And then another question you can also ask is how many F2 mutants actually bulked to increase the resolution. And you can use a simple formula, or you can do a continuous simulation, because now this number of mutants and sequence depths are going to come into play. And then the paper has a supplementary file to show what they did. And then in other frequency-based approach, the basic frequency half-half actually come into play a lot of times. You think about the bulk segregant analysis, the earlier paper that published long time ago in PNAS, there's a frequency there. And if you look at some of the later publications about this show map, I don't know if you hear about this show map, or next generation mapping, or bulk segregant RNA-seq. So the first two are pretty much our Dobs's publication. You can search online. They have nice diagram to show what is going on. It's also calculated index. It's all frequency-based approach. And then this last one, bulk the segregant analysis RNA-seq, is another recent publication from Pash-Nabel's group, where instead of bulking sequence of DNAs, you can bulk sequence RNAs. And if the gene of your interest happen to be expressed, you have to choose the right tissue, and you can not only map this gene, but you have the expression pattern information also there. So we'll stop there on the frequency on this first example. We all know that a lot of GWAS has been conducted now, started or in plants. If you look at humans, there's thousands of. But in GWAS in plants, it's still probably limited. If you talk about genome-wide association studies, if you want to compare your result with humans, even you consider your LDDK, genome size, I would consider anything with at least 100,000 SNPs, genome-wide. Of course, you can consider a QTL mapping, 100 SSR genome-wide, but because now here we are doing association studies. We do want to have large enough SNPs to capture those LD informations that to be more relevant. And then we know we are using this diverse set of germplasm. And then because of this, we are getting out of the comfortable zone of the F2s and reals. Allele frequencies are not 50-50 anymore. They were very tight relationship between the allele effect, genotypic effect, and the final R squares and variations are not there anymore. So the allele frequency is not 50-50 anymore. The detecting power we try to show here is affected by both the genetic effect, which you can define that is the difference between the two homozygous genotype, and allele frequency. Okay, In F2 and reals, we don't have that issue at all. We have everything is balanced. Even in BC populations, and you have a 50-50. If you compare the big A, and then big A, small a, and small a, you have a 50-50 of compare those two genotypes. But this, not there in the association mapping. So if your study design is used, for example, let's say you use nested association mapping as a reference parents crossed, or sometimes you can actually using different genetic design, like design 2, a diallele, and then designs other designs. And then the allele frequency, the causal mutation or polymorphism or QTN that among your founders is the impact. And then your whole, nested associated mapping panels. The frequency is very critical. And also because you typically we have a common parents. So the diverse founders and parents and common parents, their allele frequency is so critical in analyzing this GWAS experiment. And this is a recent study we've done in collaboration as a group of people, particularly Pat Schnabel. And then the question we asked is actually the genic, non-genic contributions to natural variation of quantitative traits in maize. And this is probably more relevant to wheat and barley, which are complex genomes. The result, our findings, seemingly, you probably know this, but this is probably one of the first experiments to validate that is the case. So I will skip all the detailed process of how we did this GWAS, because today's topic is on the frequency issues. So here, what we did is actually we did GWAS with maize NAM population. I'll show this maize NAM population next slide. For 5 quantitative traits, there's the flowering time, male flowering time, female flowering time, and then we did a 2-stage genome scan. First stage is a multi-family QTL mapping with tagged SNPs. And the second stage is targeted dissection with background control. If any of you will be using NAM type of populations or similar populations for your study, I encourage you to look into this one. This is probably the most updated analysis for NAM. And then the reason we do this is because this considers the genetic design of NAM population plus the multiple polygenic effect, or like a CIM type of analysis. Okay, so the result of this study is actually it's a tabulation of trait-associated SNPs. So these are just the GWAS signals across genome, and then partition them into different genomic annotation sets, which I'll show you next slides. So there's two conclusions we had. The first conclusion is that evolution alteration in protein sequence appear to be quantitatively less important than changes in gene regulation in shaping the wild natural variation observed in maize. In other words, we found the gene expression differences probably more prevalent in controlled QTLs than the stop codons, deletions, these other mutations that can change drastically change the protein sequence. What some explanation on this too. And then the next one is a genotyping method designed to discover SNPs in genes and the upstream regions would be the most economical approach for detecting signals in routine GWAS. Because sometimes if you compare GWAS studies, instead of in humans, a lot of GWAS that has been done, it became a routine GWAS. But in major crop species, maybe there's one large GWAS consortium was doing something. But for this GWAS to be done with multiple populations, at multiple traits, and then we could actually focus on the genes, but plus the upstream regions are probably a better deal. Okay, this is probably a familiar slide to some of you called the nested association mapping design. The first concept was introduced in one of the earlier papers in 2008 in genetics, and then there's some diagrams. I actually, wait a minute, I actually saw one of the professors in Montana State, wheat breeder, I forgot his name, and he actually did a presentation. I saw the presentation on this website about nested association mapping. It was very nicely done. And so we have this 25 Darcy line cross B73, you have 25 F1, and then you have single sig descent, you have 25 related mapping populations. We know that if you look using this common parent specific SNPs or tagging SNPs, you can map QTLs. And then if you sequence or high density SNP genotype in the founders, you can do the NAM nested association analysis. These other two papers published in Science is not the final analysis. There are other papers published in Nature Genetics that is nested association analysis. These two science papers basically structure and then genotyping and also the multi-family QTL analysis. They're not the final stage, but there's some concept introduced here too. Oh, Luther Talbert, yes. Thank you, Jamie. I was going to say that. Yeah, wonderful. The next slides, I actually put the title at the bottom, Genome-wide landscape of TES, because I want to highlight this whatever is going on. We try to show here. So on the left circle, or on what you call it, oval, and here we simply can partition the genome into non-genic, or sometimes we call intergenic, sometimes called genic. The difference is that the cut-off point is actually the TSS site, and this is the starting site and also the end of the 3' UTR. You can partition non-genic and genic, and then the hashed by here is the proportion of SNPs that we originally used for the GWAS, NAM GWAS analysis. You can see that the genic SNPs has a higher proportion. The hashed bar is higher than this left one, right? Right one higher than this one. And then the solid bar on the left is that after we done this two-stage genome scan, there's a TAS, trait associated SNPs. We have millions of SNPs did a genome scan, and then we have about let's say 100 or 300 of this TAS. And then you do a proportion, whether they're from the non-genic or from genic. You can see that this non-genics is actually the proportion that became higher. So there's an enrichment of non-genic SNPs within this TS signals, within the GWAS signals. There's a slightly depletion of the genics. You started with a lot of SNPs in the genic region, but after the test, within the winner pool, within the signal pool, there's a less proportion there. And then moving to the right side, you can show that here we further partition this genic region, became this 5' UTR, CD coding sequence, and introns, and 3' UTR. And also, within this non-genic region, you can carve out the promoter 1 kb, promoter 5 kb. And here, traditionally, when we study single genes, we think that this promoter regions belongs to the genes, right? But in the genome-wide scale, we do have to use this, what do you call this, translation start site, transcription start site, TSS, transcription start site, TSS, to separate the genome to the non-genic and genic regions. Interestingly, you can see that within this promoter 5 kb, it's a huge increase, actually enrichment of the signals versus the number of SNPs. And then within this genic region, you can see that the CDS, the coding sequence, you have a depletion. Further, if you partition the CDS into the, let's move this Okay, you can see that panel C actually. Synonymous and non-synonymous substitutions. You can see that the synonymous substitutions, this hashed bar is number of SNPs going, proportion of SNPs going to the test. Solid bar is the proportion of the winner among the winners pool. You can see the non-synonymous substitutions is actually depleted. So within the signals, they're not that many is stop codons or changing the changing the amino acid, So that's interesting. This is what we call the genome-wide landscape of TES. So there's actually the enrichment on the promoter regions in terms of the GWAS signals. Okay, here, we have the title as TS versus functional polymorphisms from QTL, this is actually typo, from QTL cloning. So on this upper panel with this green line, this is a summary of cloning of major QTLs from different species, from tomato, rice, and there's probably wheat too, and then the maize. You can say within those major QTLs, they're controlling, let's say, volatilization, domestication, shattering, or fruit size. They have a huge effect. Typically, they were done with the QTL cloning from a population you cross the wild type and to the domesticated type, or from large fruit to a small fruit. And then within those major QTL cloning process, the CDS has 21 of them. Now I just put this, because this number I know I just put is actually small, I just put 21 here. And then the promoter regions, sometimes promoter or CDS is 6, sometimes promoter is 3. So you can see that if it's qualitative traits or major QTLs or the significant changes of the trait values, it's more likely you finding out the causal mutations from the CDS. But it's here, which is different. You look at the 3 red arrows here, and then promoter 5KB is actually here, and CDS is actually a little bit depleted. Again, here now is another frequency issues I like to share. This is because one of the reviewers of the manuscript actually really commented on this figure, saying that this figure has some educational functions. So we'll try to see if you can understand this. This is a 3D plot. On the x-axis is the allele frequency. On the y-axis is the genetic frequency. And then this genetic frequency is actually scaled from 0 to the minus 1.5 and to the 1.5. This is overall the 5 traits, old traits, 5 quantitative traits. And on the z-axis is a chance to detect this QTLs, and then the R-square value. So because of this NAM design, the common parents B73, its allele, it's there. So you can see that the allele frequency we just plotted from 0 to 50%. As the allele frequency increase, the chance to be detected and your R-square kind of increase, right? You can increase that. Something happened that this okay, now it's coming out again. Good. Oh, it's going away. Okay, and then genetic effect. As you deviate from these zeros to the both side, the amount of variation you explain R-square is going to be smaller. You can see this, going inside and going up. So for anything to be detected, either you have a nice frequency or you have large genetic effect. See this one you have a bad frequency, non-favorable frequency, you better have a large effect. That's why this, what do you call this shape, this micro, shape. You have to be away from this center line of zeros to be detected. Because you have bad frequency, you have large effect. But if you have a good frequency to be detected, good frequency, good contrast, you have also a large effect. Your R-squared is going to be really large, and it's easy to detect these QTLs. So the allele frequency was the genetic effect. Two factors come into play. So this is the original publication where we have this leaf length, leaf width, angle, and then date to antithesis, date to silking, and all trait combined. You can see this general tendency show up in all of this trait, and sometimes you have an extremely large effect, even have a low frequency, you have high bar here. This again the same as this last one is the same as the one we showed in last slides. So just, again, for this section of the GWAS is allele frequency and genotype frequency influence the detection power. Allele frequency among founder inbreds and genetic design determine the final allele frequency of the resulting populations. This is very relevant to the NAM type of design. If you have multiple common parents, some of them, if the gene of interest of some kind of gene is not detected, it's not saying they're wrong. It's just saying maybe have a bad frequency to be detected. Their frequency was too low to be detected. And then frequency of TES in different genomic annotation set and frequency of SNPs can be compared to answer relevant questions. That's what we did in those genic and non-genic comparisons. This is another study we did in QTL cloning. A parallel domestication of SHADRON1 gene in cereal. The reason I started this kind of pet project is that people know me for a long time. Know we are doing a lot of this, QK mix model, NAM design. This thing is always quantitative genetics, and then I just feel like I like to see those genes get cloned and show this specific sequence and gene expression, so that we do not only do quantitative genetics in formulas, detections, we can just see the genes. So this is the postdoc Zhongwei Lin did all this study. There's three major conclusions. One is SHADRON1 control SHADRON and non-SHADRON in sorghum domestication, and then frequency distribution of multiple SH1 allele agree with geographical distribution of SHADRON of sorghum races. And then this last one is a parallel domestication, because when we did this, when we found this gene in sorghum, we also found evidence in rice using a mutant line, did 1,000 and then PCR, and also it happened that in maize, there's two very strong QTLs that contain, this QTL has a confidence interval within those two narrow intervals. One has 59 genes, one has 12 genes. The SHADRON1 oscillogs are present in both of the QTL regions. Very interesting study. You can look into details. We'll just focus on the frequency issues today. So this is the second conclusion. So I skip all this QTL cloning process. So multiple domestication of sorghum, because I'm going to make a story on this frequency of multiple alleles. Here, well, let's go to the right side. Basically, this is a map of the Africa continent. And then sorghum has 5 major races. One is Guinea. It's a Western African line, but also showing up in the Eastern African highlands. And then the Dora is a North African and Equator region, or some Ethiopia. And the Caudedum is another Central African race. And Kafra is basically a southern race. The distribution is shaded by these colors. And then we actually found there's 3 domesticated alleles. So we use small SH1 because of the loss of shattering. So it's just small SH1, SC265, TX430, and then TX623. And then on the left side, this is the distribution table. And then if you see that within the germplasm we studied, the Dura race, almost 100% of them, the percentage is calculated per row, is pretty much all SC625. That's why I put this here on the map. And then the Guinea is actually majority is the SC265, which is again agreed because the Stora and the Guinea, they're close to each other. And then Kafirs, it's pretty much TX623. They're in the Southern African region. And Cadatum, because here you see a lot of things are shaded, double shaded or triple shaded here, because this area where you have multiple things together. And then you have this major, it's still TX430 alleles, but you have this TX623 and the SC265. And bicolor, the reason I did not show bicolor on the continental map of the Africa is because it's distributed everywhere, but nowhere dominance. So you can see that it has this TX623 alleles and then 265 alleles. Frequency distribution can help you to see these alleles, put these alleles on the world map. Very nice. Here, actually the gene itself. This gene is pretty large. You can see here it itself has a 2.2 kb indels in some of the lines. And on the left side is the promoter region. ATG is the starting site, and then TA site, and then this is the end of the 3' UTR. So on the first haplotype, which is allele, this is the wild type allele. There's sorghum vagetum, sorghum perpinquim, and one of the lines central deshadron, P89003. And then we actually have a total sample of 25 of them have the same haplotype at all these different polymorphisms. Some of our structural variations. And then on the other side, on the domesticated allele, you have one allele, which is the TX265 allele, another allele 436, another TX623. So the functional site, the final functional site, we validated with gene expression and with protein sequence prediction, is shown by this 3 different colors: the blue color, red color. These two we cannot separate. We just put them together. It's supposed to be an expression. And here is a deletion. It's actually a large deletion. It's truncated one of the exons, actually 2 of the exons. So, and then these 3 different alleles, this wild type allele and the 3 domesticated alleles, they have numbers of accessions or sample size listed here. And these are the protein sequence predictions. And you can see that one of the TX623 is expression difference. So that actually the protein sequence is the same. And then one is a truncation, another is a severe truncation. This is one of the zinc finger domain and the Jianming domain of the gene. And then this is the expression one. This one, TX430, has low expression. 265 have actually truncated. Which, you see this one is a 526 base pair. And then this TX623, the 316, this is actually the what do you call the cDNA sequence length. Okay. This is probably most relevant site to this talk. I showed you about 3 different function site. This is the function site. This is the edge of those 2 deletions. Second, this is another splicing site mutations. When you lump everything together to do let's say an LD plot on the lower half, or do do association test, you find out actually this function site themselves do not give the high p-values, right? Because of multiple haplotypes. And this non-functional site, this other site, because they are in LD with some of this function site, they tend out to give you higher minus log p-values. Let's say this is a minus log p equals 10. And unless finally you figure out all the alleles, you get this multiple haplotype test, you get this very significant result. In Chinese, we say there's many stars, this is the moon, right? Unless you figure out everything. And then on this LD plot, The red dot basically LD equals 1 because those are the edge of the two alleles. And then you can see this, typically this non-functional site in LD about 0.3 with the first haplotype and second haplotype and third haplotype. They combine together, it have better frequencies, have higher p-values. This slide basically show that all together. So this you see this red regions or the green regions, and they're not necessarily have the higher p-values because of the multiple allele issues. Okay. The last one we're going to touch briefly is a missing heritability or rare variance issue. And then this is probably well known by this time. Missing heritability is that when we, try to do GWAS and find those genes, and then you put them together, they explain only a small minority of the inferred genetic variance, a small proportion. Remember, in humans, this missing heritability is not QTL or QTing, they're just validated genes. There's some misunderstanding in the plant community thinking they can actually address the issue. I don't think so because we don't get to validate many of those genes. In human, they have large communities, they actually validate those GWAS result and then do this calculation on missing heritability. It's not just QTLs. And what would account for the missed heritabilities? People propose the structural variations, epistasis, G by E, heritable epigenetic factors. One of the reasons one of the things they proposed is rare variance with large effect. So, and this is one of the earlier papers in Nature about this missed heritability. And this missed heritability was actually joked as dark matters. You can feel it's there. It's you can find it, right? It's actually also like we know the trait heritability is there, but when we try to dissect this into individual QTNs, quantitative trait nucleotide, and when we try to increase resolution, we always lose the power. You don't add up together to be that high percentage of variation explained. We have a postdoc Chen Song was showing at this here, and he did some theoretical derivations. Basically, instead of just genetic effect size, small a, and also allele frequency can define as rare allele, low frequency allele, common alleles as minor allele. This MAF is minor allele frequencies, smaller than 1%, and then between 1 and 5 or above 5. He also considered this is actually very known in human genetics or population genetics, the relationship between genetic effect size and allele frequencies. So here, just basically saying that if you use a double exponential distribution, double exponential, it just try to get it symmetric. If you're talking about the common variants, lots of them have their narrow peak here. A lot of them have small effect. The density around this zero, small effect, is high. And if you talk about the rare variants, maybe they have a larger proportion, tends to have large effect. There's a lot of population genetics reasonings behind this different distributions. So instead of effect frequency, there might be some relationship between the frequency and then size, because there's an evolutionary history here. The brief summary is that in most cases, rare variants only contribute to a small proportion to the overall genetic variance of traits. That's because of the derivation, even after you consider these different distributions. And in the empirical analysis, we did this whole genome sequence of the 962 European Africans of the CHARGE consortium, and it have 25 million SNPs over 22 autosomes. 6 quantitative traits, just are like our yield and moisture flowering time. You can estimate the contribution from each allele frequency bins. So here, you would actually try to calculate this genetic relationship matrix with SNPs with a specific frequencies. So this mixed model procedures, which I not try to confuse you here. Basically, if you do things like this multiple variance component estimate, you'll be able to actually quantify the overall genetic, within this overall genetic variations, how many is different, how many can be attributed to different beings. And then this is the empirical result, the BMR and height, LDL, one is good cholesterol, one is bad cholesterol, and then total triglycerides and total, just those traits. And you can see that this is the when you have all SNPs fitted together. If you only fit common SNPs, and if you fit this common SNP, low frequency SNP, and this rare SNPs. So all this tip are the actually rare variants. You can see that rare variants explain some, variations there, and then but their overall is probably 2 to 8% of the genetic variation that explained here. Okay, we'll just because it's a very lengthy study, we'll just focus on the main topic is actually contribution of rare variants to total phenotypic variance range from 2% to 8% of the 6 human quantitative traits. This is what we estimated. They might be not the same as after you clone all those QTLs and genes or QTMs, but that's the overall estimate. And empirical detection and validation of rare and common variants would further enrich our understanding of the relationship between the effect allele frequency and the effect size. Typically, we will just remember that when we calculate the additive variance, we'll just have this two. But there might be a relationship between these two during the evolutionary history. So that's the third part. These are the references of MutMap and the genic non-genic contribution on genome research. This is a Nature Genetics paper in this QTL cloning. This is just basically showing this multiple haplotypes when you're doing testing, the simple traits, right? Not the complicated traits. This study is a manuscript being prepared. The take-home message, again, go back to the allele frequency and genotype frequency are fundamental concepts. I strongly encourage all graduate students and postdocs to review some of the basic formulas. And you don't have to be expert on any of this. Just remember that if we actually study an area out of the comfortable zone of the QTL mapping, the frequency always come into play. Even within this QTL mapping, there's frequencies here because of the recombination between the marker and the QTL, right? The frequency come into play here too. So many opportunities and challenges in the genetic and genomic analysis and plant breeding have to do with frequencies. It won't go away, basically. And this acknowledgement, the NSF funded the SAM project for the genic, non-genic study. And then Pat Snable was one of the co-corresponding author of the paper, did a lot of RNA-seq and analysis together. Mike Scallon at Cornell is the PI of the SAM group. Gary Muehlbauer contributed the original idea of looking into this genic, non-genic thing. Meyer Timmerman contributed a lot of our analysis and interpretations. Tesfeld Teso is a sorghum breeder. Mitch was a previous breeder, and Frank White is very instrumental in having this targeted excellence program established in sorghum at K-State, and then have our funding to do this shattering cloning.
Some more questions?
Xiaoliu Ming was one of the human geneticists. We work on this chart consortium data. John Dobley contributed some of the maize mapping result to validate our sorghum genes. Thank you very much. Oh, yeah, thank you. Oh, why I put Gary Muehlbauer at Cornell? It's University of Minnesota. Sorry. Thank you. Any questions? Thank you, Jean-Luc. It's great. I heard that you always showing up at these talks, and I checked with Jeff. Okay, good.
Uh, they could they could only chat. So yeah.
Oh, Jean-Luc is asking the question. when will the study on allele frequency
I saw one that's
contributions be
typing, but
available?
it didn't show up.
Were
So.
you mentioning that the human,
Oh, here's one, Jianming, from
study?
Sylvana Asanga.
Oh, yeah, we are, you know, let's go back to this. This zoo ETL, we're just wrapping the manuscript together. But you know, essentially, you know, we talked to other people. You know, it just the mixed model procedures, so that there's something to do with uh, because now you're calculating. Let's go back to this slides. Uh, Here you use the all SNPs to do the genetic relationship estimate. You use common here. Here you use three. You partition the SNP into three categories. You can further partition the common SNPs into different bins. Within each partition, you have to do some justification of the LD before you calculate this genetic relationship matrix, and then feed this all components together. This actually, you know, multiple various component estimate together. Then you can, you know, partition that. This is just, we're still wrapping up this result. Thank you. Oh, thank you, Sarah. I actually expecting students to ask them questions, make this useful talk. Can you raise, Jeff, can you raise hand and then communicate by voice? Oh, you'll choose to chat. Okay, it's okay. Yeah. Okay, it's fine. In calculation of allele frequency, do you assume the allele interaction are known? Uh, in calculation of allele frequency, were you thinking that, were you asking a question about this allele, rare allele contributions to quantitative genetic variance? Okay, let me address this question like this one. We know that there's some overlap between different alleles in different allele frequency bins. When we try to map them together, you know, those overlapping parts could, you know, just have to be ignored. Yeah, you know, allelic interaction unknown. It just if you can separate from, let's say, some effect, some variations, you cannot separate whether this, you cannot, you're not sure whether it's contributed from rare alleles or the common alleles because of the LD between those two categories. You can't do anything about that. Yeah. I don't know if you have another question, but then Jean-Luc said you mentioned this, but maybe more details. Calculate multiple relationship matrix with loci different microarray, then expanding multiple. Yes, I think that interpretation is correct. Oh, that's okay. Yeah, I'm still staying on these slides. It's actually interesting that if you feed this all